Welcome to the Cannabis Growth, Manufacturing, and Testing Module Section 3, the Cannabis Harvest Process. Cannabinoids and terpenes are predominantly found in the trichomes of cannabis plants. The ratio of different cannabinoids are genetically determined, but potency can be influenced by plant age and environmental factors such as light, temperature, and humidity. Cannabinoid and terpenoid content reaches a maximum when the cannabis inflorescences are fully mature. Physical evaluation of the cannabis flowers can be used to determine maturity. Typically, a flower is mature when 75 to 95% of stigmas turn reddish-brown and shrivel, firm resistance of the inflorescence is felt when pressed, and trichomes turn cloudy. New trichomes are crystal clear, but turn cloudy and then yellow-brown with age. Trichome clouding is often associated with peak cannabis potency, but research has shown this is not always an accurate estimation. Analytical techniques such as high-performance liquid chromatography or gas chromatography are the most appropriate and accurate methods for determining optimal cannabis harvest times. There are two primary ways to harvest mature cannabis inflorescences. The first is to harvest the individual buds or branches as they ripen. This method allows for removal of the upper buds first, while allowing lower branches more time to develop buds previously hidden underneath the canopy. Typically, the stem is clipped from the plant first, and then individual buds are clipped from the stems. Large leaves are then removed from the bud, followed by the removal of small leaves. The other way to harvest cannabis is to harvest the entire plant at once. The plant is clipped and hung upside down in a drying room. The large leaves are then removed while the plant is hung. Bud trimming and clipping then proceeds as usual. After harvest, dead leaves and stems and the leaves immediately surrounding the bud are removed in a process called manicuring or trimming. This can occur when flowers are fresh or dried and can be done by hand, machine, or a combination of both. Trimming is often done over a screen to allow for the collection of any trichomes that may fall off during the manufacturing process. These loose trichomes, known as keef, have little vegetation and contain high concentrations of cannabinoids. The small, multi-fingered leaves surrounding the inflorescence contain a higher concentration of cannabinoids than larger leaves and can also be used to produce medicinal preparations. Some growers keep these leaves intact to protect the trichomes during storage of the bud until the material is to be used. Manicuring should be done under cool temperatures with good air circulation to prevent molding of the cannabis buds. After harvest, the inflorescences are dried to a moisture content of about 10%. Drying is usually done by cutting the flowering tops off and placing them in boxes, trays, or screens, or by hanging the plant upside down in a shaded area. Initial drying typically occurs over 3 days at a temperature of 59 to 68 degrees Fahrenheit and a humidity of 35% until flowers reach 25% of their original weight. At this point, flowers are often placed into plastic bags, paper bags, or glass containers for the final drying process. Care must be taken to dry inflorescences properly, both to preserve the cannabinoids and terpenes, as well as to prevent mold growth as the plant dries. Fans, dehumidifiers, and heaters are often used to control the drying environment. Once cannabis is properly dried, degradation of cannabinoids is negligible if protected from air and light. THC is especially sensitive to degradation by oxygen and light, and decarboxylation of THCA to delta-9 THC may occur with exposure, followed by further degradation to CBN over time. Short-term storage should be at 64 to 68 degrees Fahrenheit. For long-term storage, freezing cannabis at -4 degrees Fahrenheit is recommended. Cannabis material should be free of stems larger than 1.5 centimeters, and only subtending leaves should be present. The color should be consistent, with no signs of gray or black, which are indicators of fungal infection. Material should be free of molds and bacteria with a high level of pathogenicity, such as E. coli or Aspergillus, and free from heavy metals and pesticides or fungicides that can present a health hazard to the consumer. Cannabis supplied by dispensaries should be fully traceable, with a verifiable chain of custody disclosing the plant type, method of cultivation, and location of the grow. This information should be made available to patients and practitioners upon request. The Cannabis Committee of the American Herbal Products Association has developed a set of draft guidelines outlining recommended practices for dispensaries. It is important to note that while good agricultural practices do exist in the cannabis industry, not all cultivators implement them properly. Outdoor cultivation, particularly illicit cultivation, is associated with the illegal diversion of water, the clear-cutting of trees, dumping of chemicals, misappropriation of state and federal lands, and the disruption of sensitive ecosystems. Indoor cultivation is associated with high carbon emissions. The energy-intensive processes associated with all aspects of an indoor cannabis grow are estimated to consume about 1% of the national electricity use.